Okay. So it's a pleasure to welcome Professor Rabadan today to ISI USC. Professor Rabadan is the Gerald and Janet Cyrus Professor in the Departments of System Biology, Biomedical Informatics, and Surgery at Columbia University. He is director of the program for mathematical genomics, and he was director of the NCI Center for Topology of Cancer Evolution and Heterogeneity, also at Columbia. He follows like a great tradition of physicists that move into system biology and health sciences. He received a PhD in physics from the Universidad Autónoma de Madrid, Autonomous University of Madrid. He was a fellow in theoretical physics at the European Laboratory for Particle Physics, CERN, as you may know. And he also, and from there he moved to the Institute for Advanced Study at Princeton, and finally to Columbia University. I also would like to thank the Instituto Cervantes at Los Angeles and its cultural director, Gonzalo del Puerto, who facilitated this talk. For those that don't know, the Instituto Cervantes is a Spanish cultural institution that promotes Spanish culture and language. And it recently was established in Los Angeles, and they have many activities, so check them out. And today, Professor Rabadan is going to talk us about foundation models for cancer. So please join me in welcoming Professor Rabadan.
Very much for the invitation. Thank you, Gonzalo, for this nice invitation. I'm going to be talking, about some of the work that we are doing at Columbia, but also with colleagues in many places in the United States, also people in Europe. And it's a problem that is still open, and we don't know exactly what is the best way to attack it. There are new tools coming every day, but it's very exciting. It's very exciting because something interesting and probably very useful is coming from there. So how do I understand cancer? What is the origin of cancer and how cancers evolve? So cancer is a disease of our own cells. There are mutations in our own cells. We have 10 to the 14 cells in our body, and these mutations constantly accumulate mutations. We have tons, every cell in our body has mutations. If the mutations happen in the wrong or the right gene, these cells can start proliferating, and then you get the clonal process. Clonal means that it's coming from the same cell and is growing. And these mutations continue to accumulate. These cells can start growing. They start replicating faster. And then at some point, the tumors can be diagnosed, and then you get many mutations. The other mutations that we have, they are the ones that are inherited. They're from our parents. They're called germline mutations. And this composition of germline and somatic mutations is usually the landscape of mutations that we can find in cancer patients. Germline mutations, they are associated usually to risk. I was in a conference, for example, last week in Toronto, and they were looking at families that they have cancers. The kids, they get cancers when they are 4 years old, another cancer when they are 7 years old, another one when they are 10. Many of those, they don't survive very long. And they're coming in families, and families, they can have mutations. In this case, for example, it was in P53, and they are called different mini families, that they're the doctors who discovered this disease. And you can follow these families, and they are germline mutations. These are very high penetrant mutations, saying that if the probability of getting a cancer once you get the mutation is very high. But there are also many, and the genetic makeup that we have gives predisposition to certain cancers. And there have been long studies that trying to understand what are the regions in the genome or what are the different mutations that can contribute to this risk of different cancers. And this is from the germline. And then, but most of the work of my group and most of the groups in working on cancer genomics have been in trying to understand somatic mutations. And they are the ones that they are developing in our lifetime. And mutations happened, for many reasons as we age. This is most of the mutations in many cancers is as we age. They're biological processes that accumulate mutations. And they accumulate with a rate. Sometimes you can use it as a clock. So you see, for example, sometimes you have a cancer from a kid and cancer for an old person, and you can see exactly the number of mutations correlate very nicely with age, certain types of mutations. Other ones, if you smoke, you have several mutations that are accumulated because of this smoking or UV radiation, or many different things like DNA repair genes that can be altered and other things. So usually if we want to study this type of mutations, the ones that are somatic, we use a procedure where we take a sample from the cancer and a sample from normal tissue, sometimes blood, and then we sequence the genome of one, we sequence the genome of the other, and we compare, and these are the mutations that accumulate in the lifetime of this person, or this cancer. But there are many problems that are open here, and I'm going to mention some of those. The problem is much more complex than just mutations. Now we know the immune microenvironment plays an important role. Many other cells, they are still interacting with that. It's not only just the cancer cell that is important, but it's an ecosystem. And then we want to understand the whole ecosystem, cancer as an ecosystem. But since 2008 to 2009, people have been sequencing many of these tumors. This is one of the first papers that came through this kind of work, and what I call it the cartography of tumor mutations. These are studies that are called cross-sectional. And the idea is that you take, for example, cancers with, say, a leukemia, and then you have hundreds of patients with this cancer. You look at the somatic mutations, the ones that appear in the lifetime of a person, of one patient, or another patient, of another patient, of another patient. And then you try to look at the distribution. And one of the things that you observe is that the mutations, they're all different. I mean, in most of the genes, the mutations, they are going to be different. There are only a few mutations that are in common. And using some statistical procedures, you can try to see if these mutations in common, they have a functional impact. And then now we have hundreds of thousands of this cancer sequence, and now the catalogs are even larger. But one of the things that you realize here, this is one of the first analyses that I was mentioning, there are some tumors, for example, on the left, that they have very few mutations. Each of the dots is different patient, a different number of mutations per megabase of the different patients, so just a surrogate of the total number of mutations that they have. And then you realize that each of the dots is a different patient. And then there are cancers, for example, the ones there, the pediatric cancers, airway sarcoma, AML, they have less mutations. That's something that we know that is associated with some exposures, melanoma, UV radiation, or lung, different type of lung cancer that is associated with exposure to tobacco products. And then this is what we call the standard model of cancer evolution.
Okay.
Yes.
So that means that some cancers really need fewer mutations to develop, right? So that's right. The idea.
There are some cancers, usually pediatric cancer. There's a very nice observation. I can tell you an historical. I need the whiteboard also because I also, when I'm teaching these things here, but the people cannot see. So in 1953, there was a very interesting paper from a statistician, Nortley. He was looking at the number of people who died from cancer in different countries. And then something that we know, that pediatric cancers, they are rare. And people who are young is rare. And as you increase with age, the risk of many cancers start increasing. So this guy was counting, he wants to all the registries of deaths. I mean, the time was deaths of different cancers. And was plotting a curve, and it was in a log scale, the number of cases, and it was a straight line. It was increasing in a polynomial fashion in each of these countries. It was exactly the same thing. So it was the same law was saying that there was the number of people who die from cancer in all the different countries is basically the same in the proportion of the population of the country. And the idea that it was developed at the time is that mutations accumulate with age. As you get more mutations, you are more likely to get it. So tumors usually when you're younger, it's more unlikely that you are going to get the right combination of mutations. As you are getting older, the tumors, they get more mutations and you are more exposed to all these other things. And then the likelihood of developing cancer is higher.
So those cancers will be more like inherited, I mean, like with inherited mutations, like somatic.
That's a very good point.
For example,
this is a very good point. These are all somatic. But there are families, for example, in the National Cancer Institute in Madrid, there are groups of very good groups of people working with families. They look at families that they get cancers. And usually these cancers, like I was mentioning with the leaf-mini, happen in younger population, and they have fewer mutations. There are other cancers also that we are working on. This is another talk that I have here, have to do with cancers and viruses. There are certain viruses that cause cancer. And there are 7 viruses that have been found at the moment. And these cancers, they have very few mutations because they have an extra agent that contributes basically with the background of mutations that is coming through the virus. So this is true, and then it reflects basically the biology of the project and of the cancer, of the cell of origin, but also he's saying also something about the different exogenous factors that they are contributing. But usually, if you plot, it depends on the signature. This is reflected on the lower panel, but this will take another time to do that. But some of these mutations, they increase with age. Basically, what you get in any cell, you can do, I call it archaeology of that. You sequence a patient, you take a cancer, you sequence the patient, you look at the mutations, And then you can decompose the mutations in different flavors. They are called signatures. Some of those, they are associated with age, as we get old. Other ones, they are associated with UV radiation, with different tobacco compounds, different exposures of different kinds. So you can decompose the mutations in different things. Usually junior, I mean, the people, the pediatric tumors, they have very few mutations, but they are very high penetrance. So they are very interesting mutations.
And are these some, sorry, it's like a fascinating slide. So I see that melanoma is more C to T kind of changes. Is that because maybe that's a preferred pathway for like?
So this is called
ultraviolet.
So again,
ultraviolet.
These things. There's the flavor of mutations. You can catalog the flavor of mutations. So UV radiation creates a particular change in the bases with different probabilities. So you can take a base that is changed and the surrounding bases around, and then give you kind of a pattern. And then you look where it is changing. And these patterns you can catalog. It depends on if you are UV radiation, you have a specific type of mutations. If you have different tobacco has a different flavor of different mutations. These are called mutational signatures. And there are many people looking into that. They're classifying, by the way, it's a nice computational problem because you're looking of kind of orthogonal signals. They are kind of contributing to the different mutations that happen in different patients. Some of those we know the etiology, we know what is causing that. Half of those we don't know what is causing that. So probably other things that we are drinking or we're breathing or something else contributing to some of the mutations, but we don't know what they are. Interesting problem for people doing these kind of things. Okay. So here's this is the other type of study that you can do. That this is the one that I've been specialized. That they are longitudinal studies. They're things that happen with time. As I was mentioning, we have, say, 10 to the 14 cells. They are kind of constantly replicated in some tissues. And then here, time is running in this plot from left to right. And if there is a particular cell that they start acquiring some mutations, these cells start proliferating, and then you have this kind of gray thing that is kind of the daughter cells of this cell that start proliferating. Then there could be a subclone here that is in kind of orange, or I'm not sure what is the color here. And then another subclone, another granddaughter cell that is expanding there a little bit more. And then the tumor is big enough to have some symptoms, and the patient goes to the doctor, and then the tumor is diagnosed. Then the person receives different types of therapies. There large collection of different therapies, could be radiotherapy, chemotherapy, immunotherapies, depends on the tumor type. And in many cases, the therapy is successful, and then you stop the process there. Fine. In many cases, that's not the case. And then one of the other cells doesn't have to be coming from the big cell, can be another one, the blue one, survives the therapy, becomes resistant to the therapy, and start populating the next phase of the tumor development. And then you have that the tumor appears in the same site, or it can be kind of appearing in other sites, and then it's metastasis, and then you can start getting the tumor cells traveling around the body. So what this is kind of a process that is dynamic. And we have time, we have a person, we have a project that we have hundreds of people where we follow these trajectories of how these different mutations appear or not disappear. And then what we are doing is taking genomic pictures of that. We take the genome at diagnosis, so when the person was first diagnosed, compared to the normal, usually from blood or sometimes other tissue. And there we know that the mutations that appear in the first phase. And it's telling things about what are the mutations that are needed for the initiation, the oncogenesis, the beginning of the process, okay, the first diagnosis. Then, if the tumor relapses at some point, and then you can take samples also from the tumor that has relapsed. And then it's telling you something about this blue, this thing that survived the therapy. And then you can learn not only things about what started, but things about what happened to this clone that was able to survive therapy. Can inform about the mechanisms of resistance to therapy. Then you can have another therapy that is here, and then sometimes you have many lines of therapy. And then you can see how these clones, they are navigating the therapeutic landscape. Or you can be interested in understanding, for example, how different tumors metastasize, how they are able to go from the site of origin and go into different places. And for doing that, you can also sequence and trying to see if there is something in the genome that has changed between the first cell that appeared and to the cell that was able to travel to other places and start populating. questions here, the cartoons. So, and then we have these kind of longitudinal maps. I've been working mostly on brain tumors, that is on the left, but we identified many mutations. And then also in pediatric leukemias, we have a lot of papers also in this direction. So this is, for example, on the left, and this is a summary that is kind of a representation of the mutations that we can find in different patients. There are, I think, 100 circles. They're all concentrical circles here. Each of the circles represent a different patient. It represents the data from a different patient. On the axial axis, on the angle, represents the genomic position, where the mutations are found in the genome. You can say chromosome 1, chromosome 2, chromosome blah, blah, blah. And you can see that, I mean, if you just take these 100 patients, you find that there are places where you can find, I mean, most of these things do get quite random, but once you start doing some statistical analysis, there are patterns here and there. There are certain places in chromosome 7, in chromosome 17, they are very well-known genes that are associated to the progression of brain tumors. And then these are genes that we found that they're associated with pediatric leukemias to the first diagnosis and then to the relapse and mechanisms of relapse on this. Published in several of these papers, we have publications. Of these genes, we have publication. But this is only just part of the problem. So all the analysis that most people have been doing here, and this is not my work, and all the people that we've been doing here, all this work, has been looking only at a particular part of the genome. So the genome, only 1% of that is coding, is carrying information about the proteins, that is coding information. The rest is in regions that we don't know very well how to interpret. They're the non-coding parts of the genome. And most of them, or most of the technologies that we are doing, that they are used in hospitals, and they don't look at this part of the genome, except for some particular places. And the main reason is because we don't know very well how to interpret what are these the functional role. If we have something in a protein, we can try to see what the protein is doing. We can understand what is the functionality of this particular mutation. We can see how this protein is interacting with other proteins. We can have an experiment which can introduce the mutation and trying to see if the cells proliferate more or do they metastasize more or migrate more. There are different kind of phenotypes that can be associated with these things. But when we start looking in the 99% of the genome that doesn't encode information about proteins, and then we don't know very much what to do. But in the last few years, they have been very interesting analysis, but saying that there's a lot of interesting information there. In the first pair of papers, there was a couple of papers in Science in 2013. There was in a gene that is encoding the reverse transcriptase of the telomerase, that is the gene that is doing the ends of the chromosomes, that they happened in a non-coding part of the genome, in the promoter, in the very beginning of the gene, that is activating this particular gene. And this found across many different tumors, in melanomas, in gliomas, in bladder tumors, in hepatocellular carcinomas, in liver, and other ones. There's also, couple of years later, mutations in acute lymphoblastic leukemias. This is a type of leukemias in kids, in a particular region, a non-coding region of an oncogene that activates, creates a new region and activates this particular part of the genome. There was also in another transcription factor that I'm going to mention later, in PAX5, also found there. And then people have been looking at what can be the regions that are interesting for these cancers, but it has been very biased because this is looking in genes that we know that they're important, and then we look around the genes that are important, and then from there we can try to see if there is something that we can interpret. But once you go beyond these few genes that we know that they're important, there are basically very few methods to go beyond. The other type of approaches that started like a couple of years ago, 3-4 years ago, there, when you take cross-sectional data, many patients, you take the whole genome, and then you look at places where you can find more mutations than expected. So you do some statistics, you take do the whole genome, just count the number of mutations. You could say, well, I should expect in this region for these patients 3 mutations. I found 30. I suspect there is something important. And people have been trying to look at this type of mutations and trying to get, yes, these landscapes of mutations in non-coding parts of the genome. I'm going to mention a couple of those. And then, yes, I mean, this is some of the work that some of my colleagues at Columbia, they have been doing in this direction. Any questions here? I don't know if this, I'm becoming a little bit too technical or No, that's good. Good. Okay, fantastic. So, but to solve this problem, and this is a very nice kind of, I would say, interesting problem, is that we have to understand what is the mutation doing. For doing that, we need to see what is this mutation, what gene is affected, because if it's coding, we know it's in the same gene, we know that. But if it's in a part that is not coding, we don't know exactly how this relation works. And also what is important is what is the cell type. So one of the things, for example, that talking about differominics, these families that they get tons of cancer when they're kids, and they're getting, there was this cases, there was a person who has 20 cancers when they were like 40 years old. So, but they are not happening in every cell. These mutations are in every cell, but the cancers only appear in certain in sarcomas, or they appear in breast cancer. They appear only in particular cell types. So if you want to understand what is the impact of mutations in the genome, what you have to do is looking at the particular cell type. And a CD40 cell can be different from an astrocyte. It can be different from a macrophage. All these different cells, they are different. They have a different makeup. And the mutation, if you want to understand the mutation, you have to go to that particular cell and do the experiment. So experimentally, this is very complex, because experimentally you can CRISPR in the mutations. You find the mutation. You just introduce the mutation. But then you are doing in this particular cell line. If you want to do it in a different system, that's a little bit more complex, because the result that you get is there. It can be positive or it can be negative, but doesn't tell you anything about other things that are beyond that. So the problem that we are trying to do is solving this triangle, the mystery of the connection between genes, variants, and cell types, and how these are connected. Okay. Questions here?
I'll wait for it.
Okay. So this is kind of a set of tools that we are developing, other people are developing, many people are developing at the time, that is using the genome as a language. Okay, so they are kind of these bases, they're A, C, Gs, and Ts, and then you can find long phrases, and these phrases carry some information. And then in the same way that in language models, you can start asking questions about what is the meaning of some words, or what is the probability of a particular word in a particular context. Here, you can do the same thing here with these foundation models, with these non-coding parts of the genome. So this is some work I'm going to mention. That is work that was published last year by mostly by Xifu, that just graduated now from Columbia, and some of my experimental colleagues also here. That is this model cat. So what are we looking for if we want to solve this issue of what are the non-coding mutations in the genome? We're looking for methods that are able to infer transcriptional activities, meaning the amount of RNA that is generated, that is created by each gene. Okay? But this cell type is specific. So if you have a CD40 cell, it's going to be different from a CD80 cell, and it's different from a T-Rex cell. That is generalizable. So we learn in a corpus of data, and we want to learn how to transfer this data to a different condition. We want interpretability as much as we can, and we want methods that are quantitative, so that we can predict the expression of certain genes. Okay. So this is the metaphor that I'm going to use here, is like language models. Okay. So just on a nutshell, this is the first model that we have. We have another ones that they are coming very soon. We are learning two functions. Okay. The first function is what we call the grammar. Is connecting information that is the genomic information. So you have the genome of the human genome, the genome of a particular patient, and then epigenetic information. So information that is telling you what regions they can be open for transcription, that they can be activated or deactivated. This is where the information about different cell types appear. So if you look at the macrophage M1, you have a particular regions that are open. If you look at the CD40 cell, it's different. If you look at oligodendrocytes, it's a different. So each of the different cells, they have different peaks and different structures. Okay. So all these things, what we do is that we take a region of a particular size, it's around 2 megabases. We just look at regions that are open for a particular cell types, there are these peaks. And there we look at different transcription factors that they can bind, perhaps a little bit technical here. So from there, we generate a big matrix. And then we do a process where we hide some information, we mask some information, and predict what is the combination of different transcription factors that can work there. The second function that we learn, what we call this F, is the fine-tune function, is that when we have this grammar, what is the combination of different factors in a particular cell type, we can predict what is the expression, if a gene is going up or down. Once we learn these two things, we can do experiments in silico. Okay, so we can try to see what we win if we delete an enhancer, or we add a transcription factor, or we do many different things. For the first version of the model, now we have the second version, hopefully coming soon. It was trained in these 1.3 million cells from all human, adult tissues, and also fetal tissues. But we have this type of data of expression and single cell data, expression and attack data, regions that are open or closed for chromatin. So the idea there is that we have this corpus of data that we are taking many cells, and we learn what different combinations of different transcription factors and genomic information and genetic information are predicting the expression in different cell types. So just to give you a metaphor, I mean, kind of a big metaphor. If you are looking at, for example, in language models, you have a dictionary of different words that you can use. I mean, you can see, tax, tool, lawyer, DNA. I have a distribution of words that I'm using here. And then from this dictionary, you have different words that come in different domains. So for example, if this talk is about molecular biology or cancer, I use the word cancer, I use the word DNA, attack data, a gene. There are certain distributions of different words that appear in certain contexts. Okay. But if you are going to a talk with lawyers across the street, perhaps it's tax or suing someone, or doing something else. I mean, it's going to be different distributions in different contexts. Or if you are going to the Cervantes, probably there are different distributions. So each context have different distributions of words. So you learn in a corpus of data, and you learn what are the distributions of words in certain contexts. So you take a lot of data. In this case, I mean, you can take the whole, I don't know, part of the internet, the library, and then you learn distributions of these different words. Then once you have it, you can fine-tune that for particular purposes. I mean, you can say, well, how reliable is this information? How consistent, or different things. So in our model, this kind of loose metaphor, the dictionary, they are going to be different parts of the genome that they are important. What we think that they are the most important for predicting expression. We can go to a specific gene. We can look to the region that is open, for example, the promoter, and we can say that the transcription factor can be binding there. Okay, so that's one of the particular words. These are many different tokens, different words, and then they are established in different domains. So as I was mentioning, if you take an oligodendrocyte, it's going to be different from another cell that is a CD40 cell or megakaryocyte. I mean, they are different cells. They have different kind of distributions of different regions that are open or closed. And from there, you can just learn all these different things. This is what we call the regulatory grammar that appear there. And you can learn, for example, expression of many other functions that you can do. Okay. So this allows you to have a system in silico where you learn all these different grammar or how this different combination of different factors. And you can try to see if they generalize well, if they can, you can learn things about the system that you never seen before. So in this case, for example, there are several analyses that usually we do, is that you learn in, let's say, 200 cells, but you never learn, in this case, for example, is an astrocytes, so a type of cell in the brain. So the data have never seen astrocytes. Now you want to predict something in astrocytes, and then you have the prediction here on the x-axis, and then the results. And you can see that there is a nice correlation. So the system is able to recapitulate something that had never seen before. I mean, so once you have that, and this is kind of the big programmatic effort that we have, is that we can start what this doing is what we call functionalizing the whole cancer genome. Okay? So the idea is that there is a patient coming with certain mutations. 1% of the mutations, we have technology for knowing what they are doing in the protein structure, doing how functionally it can be done. The other 99%, we have to use some of these techniques. So for doing that, we kind of learn this corpus of data, all this single cell data, this telling you about how the grammar of different parts of the genome, they are correlated. And then what you do is just, you take fine-tune, you take data specific for these particular tumors. So we are working with different types of tumors and different collaborators. So these are different groups in mostly at Columbia University, but they are also in other places. So we're working in lymphomas, in acute myelogenous leukemias, in prostate cancer, DNA repair genes. There are many different things. Once you have this, it's like a language model where you can start asking questions in different contexts. So you take data from different patients, you fine tune the system, and you can do that. So let me tell you something, how this is done. So you have your data that you learn, the platform one, the base. Okay, so this is what you learn, all this grammar. And then let's say you have patients from brain tumors. I'm going to show you something about brain tumors. So you take one patient, and then you try to see how this grammar is changing in this patient, in this tumor. And then these things can be rewired, and then you see this rewired. And then from there, you start extrapolating to other patients. Okay? And then you have this type of predictions that are across patients. This is data for 16 patients of the most common type of brain tumors, glioblastomas multiforme, astrocytomas of grade 4. There are 16 patients. What you do is just, you can try to predict what is the expression of different genes. This is from single cell data in the tumor cells, in the cells that are in the tumor itself. In the macrophages, there are cells that are immune cells that are around, and other type of cells that we have there. And when you try to learn how this expression works, this is what is called zero shot. If you've never seen a patient before of this category, and then the prediction, this is the correlation. So, I mean, 1 means perfect, 0 means that there is no correlation, and this is okay. But once you have one patient, information about one patient, the performance starts increasing a lot. So you start learning what's happening, what are the process of rewiring all these systems once the cancer starts appearing. So now the interpretation, and this is another part that this could be much more complex. We try different methods. One of the methods that we use is basically how much the output changed by changing the input. So Jacobian. So if you were just an analogy, also kind of a metaphor, if you are classifying images and you are saying, well, you have a lot of images, and then you want to know what is the label here. You are trying to predict the label. You train one of these models, and then you can go back and trying to see, well, in this picture, what is more informative about these labels here, about these 3 lions, or about the dog here? And then you see that the information that is more relevant for the classification is in certain parts of the picture. So similar thing we can do. We can just go learn something that is happening in a particular patient, then going back and say what part of the genome they are going to be more important for this regulation that happens there. Okay. So these are very open models. So and then you can start asking many different questions. And this is some of the questions that I'm going to describe here. There are some of the questions that we are working at the moment, but there are many different questions, not only in cancer, as you can imagine. I mean, there are applications there are colleagues that are very much interested in other neuroscience and other different domains. But my work is mostly on cancer. So here and there are 2 different applications for cancer genomics. The first one that I'm going to spend more time is just trying to assess what are these mutations, these 99% of the mutations that we don't understand in the genome, how we can learn what could be the functional impact of these mutations. Okay. Both somatic, so mutations that are in the lifetime of a person, and mutations that are inherited from. The other one is a little bit more complicated. I mean, there's combinations of different transcription factors that probably I will skip here. Okay. So I can give you one example here. The example is in gliomas, in glioblastomas, and in brain tumors. These patients usually, there's a person started doing something unusual, some problems, go to the neurologist, take an image, and they found that there is a mass in the brain. These tumors, the treatment is have not changed in the last 20 years. The standard therapy is an alkylating agent and radiation and surgery. But usually these tumors, they come back after a year or so, and then usually the patients, they don't last very long. So there have been a lot of, there is a lot of interest trying to understand what is creating these tumors. What are the different factors? What are the risk factors of these tumors, how they evolve, and also if there are certain therapies, or how they avoid therapies. So we have been working on that for a long time. This is data that is generated by a consortium that is called GLASS. This is the glioma longitudinal analysis, so these are different time points. There are 64 patients where you have the whole genome, and you have expression. So what genes are going up or what genes are going down. And in this case, there are like 2 to 6 tumor samples per patient. That is quite unusual. So you have, when the patient is at first appear, it's usually taken, immediately taken to the neurosurgeon. The tumor is, or they extract a significant part of the tumor, and then from there you can start sequencing. So we've been involved in many processes, not in these things, but sequencing the tumors in different parts of the genome, trying to understand different parts of the tumor, trying to understand what is the history of these tumors. But the problem that we find is a problem of numbers when we are getting these mutations. So typically in a patient, you find in the coding part of the genome where they're changing the proteins is around 10 to 1,000 mutations. But if you look at the other part, the one that we don't know very well how to interpret, the numbers multiply by 100. So you get to 1,000 to like a million number of mutations that you can get. And we don't know which of the mutations are important and which are not, what can be functional and what are not. Okay. So can we use this type of models for start kind of uncovering what is the part of the genome that we cannot interpret? So, and then this is one example. Looking at a very well-known gene that has a mutation in a non-coding part of the genome that I was mentioning before, that is TERT, that this is the reverse transcriptase of the telomerase. It's a gene, it's a chromosome 5. Sorry, I can't see very well. You see them much better there. But this is, the gene is here, this part here. This is a part of the chromosome 5, and these are 2 megabases, so 2 million bases around the particular gene. And there are many other genes also here around. So if you take the cell of origin of these tumors, they're astrocytes, normal astrocytes in the brain. You can look at this normal astrocytes. This is the expression, and what genes they're expressed, they're activated. You have copies of RNA coming from there, and which are not. And you can see that there are certain genes, but the gene that we are going to be looking at is silent. In normal cells, in normal brains, in normal astrocytes, it's usually not very active. In tumors, it starts getting active. The expression is going up. So basically what you can start doing is looking at what is the part of this genome that can be linked to the activation of that particular gene. And what you see in the model, this is our model here, is that all the information is encoded in this particular region in the whole genome, in the whole region. It's 2 megabases around that. So there is a particular place there where most of the information about the expression of this gene, of TERT, is encoded. And this is coming from But then the other thing that you can do is taking these patients and count the number of mutations that appear in these patients around the particular part of the genome. Mutations happen randomly in different parts of the genome. But what you see is that in many patients, you start getting the same places, the same mutations again and again. So you found that first, there are tons of mutations. This is how they were identified in the promoter of the gene. And then what you find with these models is that these are associated to the activation of TERT. But what is interesting is that you can start doing experiments in silico. So you can start doing, you can CRISPR in the mutations, and there are tons of experiments doing that. This is if you know that they're important, but you can do the experiments in silico. So here in this on the left, you have different regions that are open in chromatin. This is the peak that I was mentioning. These are other peaks that could have some information, but very little. And these are the different transcription factors, so proteins that bind to DNA, that are associated with the increased expression of TERT. And you can see that there is a family of these particular ones. The mutations that happen, there are these 2 particular sites, the 228 and 250, that happens in many of these patients, and they are disrupting this, the binding of these proteins to the DNA in a particular way. They are creating some particular sites and disrupting other sites. This is in silico, the base editing, so in silico mutagenesis. So you can introduce these mutations, the ones that you find in patients, and you can see how they are changing the expression. And you can see that the 228, you have this increase in the expression. This one is also increasing the expression. Other ones, they're doing nothing. And this is data from the same patients where you see that these mutations, the ones that are in blue, they are the ones that are increasing the expression. Okay, this is a more complex data to analyze. So what is interesting is that now, you have tons of mutations that are found in these patients. It's very hard to know what these mutations are doing. But if you can introduce these mutations in your computer and say, well, these ones, they are going to be important, then working with my colleagues, they can do some CRISPR. They can CRISPR the ones that we're going to be important, and then we can start characterizing some of these mutations. The second example is another type of brain tumor that is also a couple of years ago, a few years ago, they were the same type of tumors. They're glioblastomas. Now they are classified into two different types. The ones that they have mutations in a particular gene that is called IDH1, that they are usually better prognosis. They are not great, but they are better prognosis. And the standard astrocytomas, grade 4. So this is another type of data that this is GWAS data. This is associated to risk. So one of the things that people realized is that these tumors, they are very rare, but they can appear in certain populations or certain people more frequently. So how do you identify what are the genomic contributors to that? So you take, let's say, 500 patients, 1,000 patients. You take 1,000 unaffected individuals from the same population. You look across the genome and you look at variants that they are common in the population and if they are enriched in the people who are getting the cancer versus the other one. So one of the things that appear very immediately is that you have, and this is called the Manhattan plot. Here is how significant is that? This is the log of minus the log of the p-value. So if something appears up, this is the whole genome from chromosome 1, 2, 3, 4 at the end. And then you see that there, and this is the red line, is something is statistically significant. And the peaks that appear here, they are regions that they are associated with higher risk of getting this particular IDH1. So you take the highest peak, this one here. And then what you see is that you have many variants here. You can see there is a region in chromosome 8 that if you inherit this region, you have much higher risk of getting this cancer. Okay, so what we can do then? We can introduce these mutations, I would say, in silico. So trying to see what are the mutations that happen, they are coming from these individuals at risk. And we're collecting information about this triple that I was mentioning before. So there is the SNP that is important, so a particular mutation. What is the gene that is acting, and what is the cell type that you are finding these things? And that was from a different paper from a colleague that introduced these mutations and saw that it was basically connecting this MYC, the activation of MYC, with this particular SNP that you introduce it, and in a particular cell type that is oligodendrocytes. What is also helping you is trying to solve something that is a super interesting problem. It's called the cell of origin. So once we see a tumor, we don't know sometimes where the tumor is coming from. What is the cell of origin? It can be, there are many different cell types in the brain. And the ones when they have the IDH1 mutations, what is the origin of that? They're astrocytes, they're neurons, what are these type of cells? So you can introduce these mutations in silico. You can understand, you can try to predict what is the effect in different cell types. And they can tell you that the effect of this particular mutation, it appears mostly in oligodendrocytes, that they are thought to be the cell of origin of these particular tumors. So the other type of analysis is that we are looking at different combinations of different transcription factors. So the cell type specificity is coming not by activation of a particular gene or a particular set of genes, but usually gangs of different genes that they are important. So if you know the cell of origin of a particular tumor, you can start doing experiments and trying to see what's going on there. And this is very briefly, these are mutations that are found in a particular transcription factor in pediatric tumors, in B-cell acute lymphoblastic leukemias. And these mutations, usually what they are doing is disrupting the interaction between the DNA and this particular protein. But there are these families that they have higher risk of getting these tumors. They have some mutations that are outside of this domain that they are not very well understood. So here, what we do, this is in, I think, figure 6 of our paper. So you can just look at what are the different factors that they're co-regulating the cell of origin, or the cell that we think is the cell of origin of these acute lymphoblastic leukemias, or B cells. And then we can try to see what are the ones that they are kind of a set of proteins that they are kind of having like interacting in these particular tumors. And one of those that we find is that there is this particular one here. And this is one is PAX5, this is DNA here. And the other one is a protein that is NR2C2 that seems to be binding there. And then we did the experiment and it was binding. And the mutation that they are found in these families, they are associated with interaction of these two proteins. And this is associated with different things. So why I'm so excited about this particular program, because I think there are many, many questions that we can start doing here. I mean, once we have these tools, I mean, and this is very much the beginning. I mean, there is Alpha Genome also that is, they published the paper a few months ago. There are many different tools that they are coming in understanding what is the impact of non-coding parts of the genome. It's not only in cancer, as I was mentioning, people working on every genetic disease. I mean, it's kind of interesting. But once you have these models, you can start asking many questions. And this is kind of what is the beauty. It's basically like a language model. That there are questions that perhaps you don't know that you have, but then in the future, you can have. And traditionally, I mean, the models that we were doing before, more kind of traditional machine learning, you have an hypothesis, you develop a method, you develop a statistics, and you have something. Here, I think it's more interesting because they are very open-ended. So sometimes you have different questions that appear, and then you can do all these different things. The first, I mean, this is a few applications. If you're interested in a particular gene that you like, you can start looking at what's happening around this gene and doing experiments. What happens if I take this transcription factor binding site, I open a genomic region, I do different things. There also the combination of different regulatory factors, so how this gang of different genes, they are kind of working in these different cell types. The impact of mutations, both somatic and germline in patients. And then how these, also not only just the tumor itself, but you can see how it's changing also the other cells that are around the tumor. But that's another topic. So I think we're almost there, isn't it? Say 59 minutes. So now what we are doing now is just we are following two different directions, kind of bigger directions. One is from the technical point of view, that is improving these models. There's the new version of this model that is hopefully coming soon. It's extended to more than 1,000 different cell types with physiological and non-physiological conditions. So cells that are exposed to radiation, or we have a project, for example, the cells that are exposed to hantavirus, and then trying to see how the virus is going there and doing different things. You can do that in many different things. They are also incorporating different types of information that it was not in the previous model. There is a huge amount of different type of epigenetic information that can be added there at different scales. But from the practical point of view, what I think is very cool is now we can have systems that we can just work with different set of patients or different settings. And I mean, I was mentioning something about brain tumors, but there are very close collaborations. Ones that I was just working today, that they are looking at stem cells. And then how with aging, I mean, the stem cells, they are changing. You can start looking at how you can apply some of these models to the process of aging in stem cells and how these things get required. The same thing with some stem cells or cells of the hematopoietic system, how they are changing with age and in cancers. And this is myelodysplasias, MDS, and acute myelogenous leukemias, but in many different directions. Pediatric cancers is something that I like. It's a topic that is super interesting because they have very few mutations in general. They are very penetrant, and some of these cases we don't know very well. There are some cancers that you cannot find many of these variants, and it's also fascinating. And some work in lymphomas, both of B-cell origin and T-cell, that we have with colleagues at Columbia. And there's some technical work here that I'm not going to discuss, but you can start introducing these mutations, and then we are working with experimentalists trying to understand what is the functional role. This is some of the work in B-cell. And well, this is the work of many people. These are the people that have been in my lab in the last 2 years or so. But mostly it's the work of Sifu, who is here. I think where is not here? This guy here, and there are some of our colleagues that now they're applying it to different tumors. And well, thank you very much for your attention. Thank you.
I should say.
The chat, there's questions in the chat. Are you able to see the chat on your computer?
Here? No.
Okay, I will read them for you. How accurate are those predictions, and how structured are the data you feed the models with?
So it depends on the prediction. So we have, I mean, for example, some of the data I was mentioning B-cell lymphomas, depends on what is the type of alteration that you do. For single nucleotide, just introducing a mutation is still not there. I mean, this is the thing, one of the things we are working there. For longer regions, I think it's quite nice. I mean, there's a whole section of the calibration within the paper about that.
Okay, the second question is, if I understand it correctly, there's information at DNA level of the patients involved in the study. Is genome going to become a common piece of information for oncologists to gather and feed this kind of system in the medical community? How likely or difficult is this for it to become part of the cancer study and control protocols?
Yes, so I think there are two different applications here. What I'm talking is about research. I mean, all these things is to discover new things of how the things are connected. For the clinical application, some of these things are very early. I mean, I will not say that this is going to be very close for clinical application. You can introduce the mutations, but it's not going to be now, I mean, that this is going to be used in the clinic. I will not go in this direction. It's interesting because sometimes you can get something that is very specific. In a complicated case, for example, it could be interesting to try to apply these models and try to see if there is any particular information that we don't know. For example, if there is cases where we don't know what is the alterations that are driving these tumors, I think it's complicated. I mean, and then you can try, but I don't think for any clinical use at the moment at all. I mean.
Okay, another question. If NLP uses byte pair encoding to build up a vocabulary of tokens from character sequences seen from data, what is a token in your case? Is it encoding single nucleotide base ATCG? Is a token codon triplets of base pairs, 64 possible, groups of codons, statistically common motifs grouped together?
No, in the model that I was describing, it was, and the basic part of information is transcription factor binding site, so if they are binding or not, and a region is open or not in attack. So basically you reduce the whole information, the tokens, they are going to be particular region and the potential binding of a transcription factor in that region. So it's not at the single nucleotide that they are mentioned there.
Okay, another question, are other variables such as lifestyle or specific events like exposure to chemicals or history of sunburns taken into account in these studies as part of the patient's history?
I don't know in the studies not in the study I was mentioning about this noncoding part of the genome, but people looking at mutational signatures, they are doing these things. They are trying to catalog all the life experiences that the person has. Even, for example, there are things about pollution. For example, if you live in a city like New York or LA versus if you live in a different place. I mean, there are people collecting all this epidemiological data. It is very, very useful. Sometimes you don't know the catalog of all the different things that can happen, but all the chemical exposures. There are, for example, colleagues there in Madrid, in Spain, who are collecting in pancreatic cancer. They are doing analysis, for example, of nails, of the microbiome, things that can tell you many things about the exposure that you can have, chemical compounds or different things. There are people collecting all this information. This information, I mean, it's not so easy to collect because you don't know what you are collecting that is going to be important or not. But there are certain things that you can get some clue about what is important or not. So there is a whole field collecting these things. I mean, exposomics, I think, is quite interesting. But not at the level of the models that I was discussing. But people who are working in kind of mutational signatures and things that can affect basically the mutations in cancer. Yes, definitely.
Okay, and we have another question. What worries you most about this approach? Example, being taken too seriously or being treated as truth versus simulation?
I think it's at the moment we are exploring. I mean, this is kind of a very exploratory phase. The results are very promising. I mean, I think there are many things that we are doing that I think they are promising. Not only just my work, but I think many of the other tools. I mean, Alpha Genome, for example, is quite good in predicting many other tracks in specific cell types. So there are many different things that I think quite promising, but it's kind of we are just in the very beginning. I don't think we should. I think we should be taking this seriously as a potential tool, but at the moment, I think we have to wait.
The add-on to that was hallucinations, misleading causality, false positives, bad data, etc.
Yes, that's all these things happen. All these things can happen. Yes.
And I have a follow-up on what you said. Let's just say you're studying pollution in a certain region. We all know how pollution affects humans, right? It still would be causal based on your genetic makeup. Not everybody who is exposed to the pollution behaves, their body doesn't behave.
Yeah, that's right. So for example, there, I mean, this is the studies of some, I mean, other people, not mine, but they've been looking in lung cancer, for example, and then looking at risk of certain lung cancers and with different mutations. Or for example, there's one very interesting example in colorectal cancer. There is this trend that happens in the last few years that many more young people are getting colorectal cancer for a reason that is not very known. So people are looking into what could be the cause of this kind of increase in colorectal cancer. It can be that there is something about the diet, there is something in the environment that we don't know. And then, so as we are collecting more information, we are getting more and more things. But what is interesting is that they are finding, for example, some particular bacteria that they're associated with a particular type of mutation. So they have a mutagen in the bacterium that they can introduce mutations in our cells, and these mutations can be linked to cancer. And this has been done in organoids, and there are a lot of papers now in this particular direction.
I would say a correlation for me would be when during the COVID pandemic, it was noted by the scientific community that different blood types responded differently to the COVID.
So in this case, for example, what you can do, I mean, first, these are all correlations in the sense that they are epidemiological studies. But then what you can do is you can do the experiment. And then, for example, the work that I was mentioning, this is a type of E. coli. So there's the standard E. coli, and there is an E. coli that has a particular gene that is called PKS. And this PKS has a mutagen site. So you have the two versions of E. coli, the ones with the mutagen and the one without the mutagen. So the experiment that they did is that you take an organoid that resembles basically some colon crypt, and then there you introduce the bacterium, you introduce the other bacterium, and then you see the mutations that are found. And then there you can just solve the causality problem in the sense that the mutations that are found in the patient, they are the same that you find here. I mean, because you can do experiments. I mean, epidemiology alone, I mean, it's usually not sufficient.
Great, thank you.
We have more questions from the audience here. Otherwise, I have a question.
I have so many questions.
Okay.
But I'll ask I wonder if you could give us a little bit of perspective. So language models and language technologies have been used to study the genome for many years, right? 2 decades probably. So what has fundamentally changed? How are these foundation models more powerful or different, or what are they able to pick up on the genome that, or maybe they just allow more scale because of their architecture?
I think it's both the scale. It's both the scale, the relationships. I mean, like for example, in epidemiology, the models, I mean, they were kind of let's say more linear in some way, or some more explicit the connection here. These connections, sometimes they are very non-obvious connections that happen there. So the models, they are more complex. The models are bigger. The data is bigger, so that you are using. So there are certain relations that they are not usually captured with a standard kind of linear approximations or just simple questions there. If you're looking for a specific question, if you're looking for something that you know, I mean, that is important, or a particular question, usually more defined models, they work okay. I mean, they work. But if they're more kind of ill-defined, asking questions and all these things, I think this approach is very, very interesting.
Any possibility to do something that's more multimodality? So, I don't know, pathology, images.
Yes, I mean, there's, this data, I mean, we are using 2 types of data, single cell attack, and this is called multiomic, I mean, attack on RNA. And then, but usually we don't have the same scale of data in other, in across many. So we have in cell lines, yes, but not in physiological conditions. In pathology, integrating all this type of data, super interesting. Yes, you can start doing. Usually they are smaller. I mean, you can just put you can start adding image there if you have image from patients in these cases or
data.
Yeah, I mean, I think you can. These are models of genomic regulation. So yeah, I would have to think, I mean, what could be the best way of adding this type of data. But in principle, it can be added in many different ways. Yes.
I think we're at time. I have like so many questions, but for the sake of time, I think we have to end now. I failed to say something that was very important in the introduction. This, Professor Rabadan is slated to be the scientific director of the National Cancer Research Institute in Spain, I think shortly. So I should probably say that if it happens. And other than that, I will wait for the meetings later to ask you
Okay,
more
fantastic.
questions about the genetics and dysregulation, all kind of things that I think are really important. But please just welcome me and thanking Professor
No, thank
Rabadan
you.
for the
Thank you
great
to you
talk.
for the invitation.
And I meant it. I really have a